All right, ladies. So last time we left off looking at the electron transfer chain. Okay, so why do we need oxygen? What's the role of the oxygen in the electron transfer chain? It's to ensure what? It's to ensure the movement of what things? Yes. It's to ensure that the electrons move from NADH or FADH2, and they ensure that they go through this system and find their way onto which molecule? Oxygen. Now, why do they go through? Because of, remember, electronegativity. So, let me ask you a simple question. In these complexes, which one is the least electronegative? First one, right? This is the least. This is the most electronegative complex. But what's the most electronegative thing in this? And it's oxygen, right? And that's why oxygen takes the electrons from the last complex, which is called cytochrome C oxidase. Now, without the oxygen, the electrons would not move. They would get stuck. So the electrons will get stuck in the chain. So if this electron can't move, then this electron can't move, and then these guys can't drop off anymore. This whole system shuts down. Okay? So I want you to understand how this works. I want you to understand. Oh, wait, hold on a second. We need to pause our recording. Okay, sorry. Where were we? Okay, yes. So, the idea is the electrons move through. That powers the pumps. You have a proton gradient, which represents potential energy. And then these guys are going to want to move from high concentration to low concentration. So they're going to want to go through this enzyme, giving this enzyme the energy to make ATP. This process is called chemiosmosis. If you remember, osmosis is what? The movement of water from towards an environment where there's a high concentration of solutes. So it's just the movement of water. But here it's the movement of a chemical, and that chemical, it's a proton, right? It's a hydrogen ion. Okay, so that's stage 4. And this is where you get most of your energy. So I'm going to ask you some questions now. What's the ratio? How much ATP do you get for every 1 NADH? So what does 1 NADH give you? How many ATP? Based on the math. Yeah, it's 3, right? You can see here, look, 2 gives you 6. 6 gives you 8. So there's a 1 to 3 ratio, right? Except for here. I'll mention this in a second, okay? Because you can see it's either 4 or 6. So there's a bit of a discrepancy there. FADH2 gives you not a 1 to 3 ratio, it gives you a 1 to 2 ratio, okay? Why? Why does FADH2 give you less energy than NADH? Think about where FADH drops. Sorry, FADH2. Take a look at where it drops its electrons. Where do you notice? So see how it actually skips this complex over here? So if you think about it, the electrons from FADH2 only activate 1, 2 pumps. Whereas the electrons from NADH activate 1, 2, 3 pumps. So NADH activates, the electrons from NADH activate more pumps, right? Which means more energy. So the reason why there's a difference there is because the electrons from NADH activate more pumps because they get dropped off earlier on. Now, there's a discrepancy here. You'll notice that the NADH can be 4 or 6 in glycolysis. In stage 2 or stage 3, it's 1 to 3. But why is it in stage 1, it's 1 to 2 or 1 to 3? Like, why is there a difference? And that actually has to do with whether or not you're bacteria or a eukaryotic cell. And I'll explain that. So, does anyone know where the mitochondria originated from? Does anyone know what initially it was? And there's a hint. It's got 2 membranes. So this is the inner membrane right here. Okay? And this is the outer membrane over here. And the mitochondria also has its own DNA. It actually has its own DNA. Because the mitochondria used to be a bacterium. It used to be a bacterium that can make energy using oxygen. What happened a long time ago is that you had a host cell, which I'm going to circle red, swallowed up a bacterium. And what happened was, as the host cell swallowed up the bacterium, like this. You guys ever study, so that's really bad drawing. Have you ever studied endocytosis? I don't know if you remember that process. That's when a cell swallows up another cell. So what happened a long time ago is that this host cell swallowed up the bacterial cell. When that happens, you get this. Oops. I need to really work on my drawings. You get something like this. You get a structure inside that looks like this. This, you can see, has 2 membranes. It has the red membrane, which represents the membrane of the host, and it has the blue membrane, which represents the membrane of the bacteria that it just swallowed up. This bacteria evolved a trick. It evolved aerobic respiration. It evolved the ability to use oxygen to make energy. So what happened a long time ago is a eukaryotic cell swallowed up an aerobic cell. They became partners, symbiotic relationship. The host cell gave the aerobic cell a home. The aerobic cell gave the host cell more energy. So they worked together. That bacterial cell eventually evolved to become the mitochondria. Okay? Now, how do we know that? Well, it has its own DNA. And if you looked at the DNA of the mitochondria, it's very bacteria-like. Okay? Because it was a bacteria. So this mitochondria used to be a free-living bacteria that was swallowed up, not to be eaten, but to be utilized in this beneficial role. So there are in other words, the reason I'm telling you this is if this was the first cell, right, that was able to carry out aerobic respiration, right? So let me draw you the bacteria cell. Now, bacteria, do they have organelles? Do they have things like mitochondria and Golgi, nuclear membrane? No, they don't, right? So if this cell, which doesn't have any organelles, figured out how to use oxygen to make energy, where would the electron transport chain have been? Sorry. Okay. Where would the electron transport chain be in a bacteria? If you have an aerobic, so if you have a bacteria that can carry out aerobic respiration, where would the electron transport chain be? We know that in a eukaryotic cell, it's in the mitochondria, and it's in the inner membrane, right? This is where it lives. And it's so throughout the inner membrane, we have the electron transport chain. Where would it be in bacteria? Has to be in a membrane. And there's only one membrane in bacteria. So where does it have to be? Has to be here, which is the cell membrane. Okay? It has to be in the cell membrane. Now, there's a reason why I'm telling you this, okay? So, here's the thing. If you look at a eukaryotic cell has, stage 1 is where? Outside the mitochondria, right? And then the products, the pyruvate, but also the NADH, have to be moved where? Inside, okay? And it costs energy to do that. Bacteria don't have that problem, do they? Because everything is happening in the same area. So you have stage 1 happening in the bacterial cytoplasm. Does it have to go anywhere? Does it have to be moved into another organelle? No. So bacteria don't have to spend energy or extra energy to move things around. So a eukaryotic cell has to spend energy. That's why you see this discrepancy. That's why you notice that it's 4 or 6. So bacteria don't have to spend any energy. So their NADH gives you the maximum amount of energy. But our cells have to spend some energy to move the NADH across, so you get less energy. Okay? That's why there's a discrepancy between these two numbers. Okay? So I just thought I'd mention that. All right. So we looked at stage 1, 2, 3, and 4. Let's talk about some modifications and regulations. All right? Let's talk about things that can affect or ways to control this. So let's talk about fermentation. This is alcohol fermentation. This is how you make wine or beer or stuff that you can't have because you're too young. Why are you laughing, Kim? All right. So stage 1. What happens in stage 1? We have glucose, and we know it becomes what? Pyruvate. Now, what happens if you have no oxygen? If you have no oxygen, we talked about you can't run this step, right? Can't run this step. Now that means that if you can't run this step, you start to accumulate this molecule right here. Because if the chain is full, if this is out of order, they cannot drop off their electrons. So they cannot become NAD+. You understand that? So what happens then is that if you go back to stage 2, stage 2 is inhibited by high levels of NADH. If NADH cannot drop off their electrons to stage 4, stage 2 will shut down because there is no NAD+ available. It's all NADH. That means this stage is shut down. If you shut down stage 2, what happens to stage 3? Can you run it? Why not? What can't you make? Can't make this. Can't make acetyl-CoA. So the logic is shut down stage 4, and then what happens is you start to accumulate high levels of NADH. The NADH feeds back, inhibits stage 2, which then shuts down stage 3. What are you left with then? Only stage 1. Okay? So fermentation is glycolysis. But it's modified glycolysis because something happens to the pyruvate. So you have all of this NADH. Now, what happens to pyruvate? Well, you can see. In alcohol fermentation, the pyruvate loses carbon, so it goes from a 3-carbon molecule to a 2-carbon molecule, because you lose carbon dioxide. And in addition, some of the NADH is regenerated back to NAD+. So the pyruvate actually becomes reduced. And the NADH becomes oxidized. Now, why do we need to regenerate some of this NADH? Why do we have to do that? Like, what's the point of making the NADH and then regenerating it back? That seems kind of stupid. Unless we look at the picture of glycolysis, which I believe I already have out here. So, let me show you this picture. This is the one that you have on your blackboard, right? Why do we need to regenerate the NADH back into NAD+? So here's glycolysis, right? Do you make energy in glycolysis? Do you get anything out of it? Yeah. It costs you 2 ATP. You can see it costs you 1 and 2, but you get back 4, right? You get back 2 over here. Remember, you got to double everything. So 2 over here and 2 over here. So you get back 4. So overall you gain how many molecules of ATP? You gain 2. So you do get energy in glycolysis, okay? But what would happen if you don't have any NAD+? So take a look at this flow chart. Do you need NAD+ anywhere? Where? You need it right here. What would happen if I cannot regenerate the NAD+? What would happen if I have a lot of NADH? You got to understand something. You got to visualize it this way. ATP and ADP and NADH and NAD+ are like teeter-totters. When you have a lot of one, you have very little of the other. So when you have a lot of ATP, you have very little ADP. When you have a lot of NADH, you have very little NAD+. So if I have a lot of NADH, that means I have very little NAD+. In fact, I may not have enough. And if I don't have enough NAD+, I can't run this step. Take a look. I need NAD+ to run this step. If I can't run this step, I got a big problem. Because now I have a pathway that is using up energy, but I'm not getting anything back. That's a big, big problem. That's a huge problem. Okay? So, I need some NADH to be converted back to NAD+, so I can run this step. That's why we need to regenerate the NADH back into NAD+. Okay? There is an enzyme that runs this step. The enzyme is called Lactate Dehydrogenase. When I first came across this enzyme, I was confused. I'm like, why is it called Lactate Dehydrogenase? I'll explain in the next series why it's called that. Because the name implies that it's taking away hydrogen. Because actually, let me explain in the next slide why it's called lactate dehydrogenase. Okay, so the enzyme that actually does this thing from NADH back to NAD+ is called lactate dehydrogenase. I'm going to come back to that in a second. Okay, so fermentation is only glycolysis. Okay, you get a lot less energy from it. In fact, if you look at the math, you only gain about 2 ATP. From the rest of respiration, you gain about another 34 to 36. So fermentation gives you about 19 times less energy than all of respiration, okay? So a lot less efficient. Here's lactic acid fermentation. This is what we do, okay? Yeast do this. We do this. It's almost exactly the same. You still have your original glucose, which becomes pyruvate. This is still the same. And then the pyruvate becomes lactic acid. The only difference between this and alcohol fermentation is there's no removal of carbon dioxide. That doesn't happen. Okay? There's no removal of carbon dioxide. But otherwise, this is still the same, right? And that enzyme I was telling you, lactate dehydrogenase, okay? That's the enzyme that runs the step. The reason why it's called lactate dehydrogenase is because, and it confused me when I came across it. I'm thinking, well, hold on. That sounds like it's taking away the hydrogen from the lactic acid. Doesn't it sound like that's what it's doing? Well, this enzyme can run this reaction the other way. So it can reverse it. And if it reverses it, then it is taking away hydrogen from lactic acid. Okay? So this enzyme can actually run this step in both directions. Okay, so that's fermentation. So here's a question. You have yeast growing in a sugar-rich medium, and they're using up 5 grams per day of the sugar when there's a pump that's working. If you turn the pump off, the glucose consumption goes up to 95 grams per day. Why? Okay, so Kim, what's the answer? Okay, so there's no oxygen, right? So if we turn the pump off, there's no oxygen. Very good. So why do we use up more sugar then? It has to work harder. So if you look at the ratio of 5 to 95, right? We have 5 grams of sugar when we have oxygen, and then we use up 95 grams of sugar without oxygen. That ratio is 1 to 19. Now, we just talked about that. Remember, normal respiration gives you how many more times energy? About 19, right? So if you don't have any oxygen, then it makes sense that if you're only using fermentation, which gives you 19 times less energy, then you have to eat 19 times more to get the same amount of energy. Okay? Here's a chart that's showing you pressure. Now, we talked about this before. In aerobic respiration, the pressure stays what? The same, or does it change? What do you think? Well, remember, now, I hope you know by now that this is not the formula for respiration. You know it's way more complicated than this, right? It's way more complicated than this very simple formula. You saw that it involves 4 stages, right? Lots of steps. So this is not aerobic respiration, but it still helps us to kind of summarize the overall reaction. So here's your sugar. We know we use up oxygen, and we produce carbon dioxide and water, which is a liquid. These two are gases, and if you look, the amount of gas you use up is equal to the amount of gas you produce. So the gas pressure will stay constant in aerobic respiration. But then see how it starts to go up here? Because if you look at alcohol fermentation, what stage is this? It's first stage. Where do we use oxygen? What stage? Do we use any oxygen in the first stage then? No. So in fermentation, you have basically, it's only stage 1. And if it's alcohol fermentation, we don't use up any oxygen in stage 1. But what do we produce? We produce gas. Okay? So the reason why the pressure goes up is because we switch to alcohol fermentation right here. And because we switch to alcohol fermentation, the pressure starts to build because you're making CO2 without using it. All right, so that's fermentation. That's a way to modify respiration. This happens when oxygen levels go what? Go really down or disappear. Let's talk about other ways that respiration can be regulated. So here's a nice little flow chart. We talked about one already. We talked about this one. When you have high levels of NADH, this enzyme, which is in stage what? Stage 2, is turned off. Remember, the other name for pyruvate decarboxylase is also known as pyruvate dehydrogenase, right? Okay, so we talked about that one. What about high levels of ATP? What do high levels of ATP inhibit? So it's this enzyme right over here, phosphofructokinase, which is really early on in glycolysis. Okay? It's actually right here. So you could see it's right over here. So why do we shut it down at that point? Why do you want to, what's happening? You have high levels of ATP. Why do we want to shut down this step? If you have a lot of this, that means you have a lot of what? You have a lot of stored what? You already have a lot of stored energy, right? So maybe you might want to take the sugar and do something else with it. Maybe you want to store it. Okay? The other way to look at it is, if I have a lot of ATP, that means I have very little what? ADP. And if I don't have enough of this, then I can't continue. Okay? So it's going to inhibit glycolysis, because I need to run this step, I'm going to need ADP. So if I have a lot of this, I should stretch that. No, that doesn't make any sense what I just said. Sorry. If you have a lot of this, you don't need ADP to run that step. You don't need ADP to run this step. You need ATP to run this step. It's fine. So just ignore what I just said. So it's because of what the previous thing. You have a lot of stored energy, so we don't want to make more energy. That would be stupid. We want to convert the energy, the sugar, into something else. Okay. What about citrate? According to this flowchart, when you have low levels of citrate, it actually speeds up. It increases respiration. Why is that? Why do low levels of citrate increase respiration? Why? So let's go back to Krebs cycle. Why do low levels of this, low levels of citrate, which is this molecule, why does it increase respiration? Well, let me ask you a question. How can I make more of this? To make this, I need more of what? This and this, right? How do I get more of this? How do I get more acetyl-CoA? By breaking down more pyruvate. But how do I get more pyruvate? By breaking down more glucose, right? So the logic is, if I have low levels of citrate, I could make more of it by breaking down more sugar, because the sugar becomes pyruvate, which becomes acetyl-CoA, and the acetyl-CoA becomes citrate. Okay? So these are just examples of ways respiration could be regulated. So I want you to understand how this works, okay? Here are some things that can affect respiration. This is not regulation. Regulation are things that the cell does on purpose, okay? These are things that the cell would encounter that's not good for the cell, okay? So for example, cyanide or carbon monoxide poisoning. Look what happens. These two molecules sit in the active site of this proton pump. The problem then is the electrons cannot move through. They get stuck. So if the electrons can't move through, what happens? What happens? If the electrons can't move, what happens to this entire process? It stops. Okay? That means the electrons get stuck. That means we start to accumulate NADH. Then the NADH shuts down stage 2, which then shuts down stage 3. What are you left with? Just stage 1, glycolysis, okay? So here's another chemical which works over here. Here's the thing. Whether it works here or here, you end up really having the same problem, okay? You can see it's in both cases the flow of electrons are going to be what? Disrupted. Okay? What about DNP? DNP is a chemical that, according to this picture, allows protons to leak through. Is that good or bad? Kimberly, why? Why is it bad? Well, where are the protons supposed to go through? Right. So we know that as these protons go through ATP synthase, you make ATP. So it's very simple logic. If they can start leaking through the membrane, that means less will go through here, which means you make less ATP. Okay? Very simple logic. And then here's a molecule called ligomycin, which actually blocks. It sits in the active site of ATP synthase and blocks the production of ATP. So maybe you'll have regular electron flow. Maybe the electrons can actually go through, but what can't you make? Can't make ATP. Can't make ATP. And then it's actually going to become a problem pumping the protons out, because where are all the protons going to get stuck? If they can't come back through, they're all going to get stuck where? Up here. And then it's going to become really hard to pump protons if they can't move back into the other side. So you won't get any ATP production because the protons can't move through. Okay, so these are things that are really bad for the cell. As you can see, they either disrupt electron flow, or they allow protons to leak through, or they prevent protons from leaking through. Not very good. I'm just going to leave off. We're going to be done in like 2 minutes. We talked about what molecule? The breakdown of what? Glucose. Does it have to be glucose? Does your body need to just use glucose for energy? No. It could turn to other things, okay? Here's a flowchart showing you how other things feed into respiration. So this shows you that we can use proteins. Okay? We see that proteins can actually go into respiration. They can be broken down and enter glycolysis or transition or Krebs cycle. Fats can also be used. If you remember, fats are made up of glycerol and fatty acid chains. And what are fatty acid chains? They're chains of what? A string of carbon. And they're pretty long, right? They can be quite long. Now, of the 3 nutrients, which one gives you the most energy? So proteins and carbohydrates, are about 4 calories per gram. Fats are 9 per gram. Fats give you way more energy. And the reason is because of their structure. Because fats have these long chains. And if you take these chains and you cut them into 2 carbon fragments, those 2 carbon fragments, where can they go? Look at the chart. Where did they go? What's acetyl-CoA? What is it? Remember, it's a 2 carbon fragment, right? So the reason why fats give you a lot of energy is because if you look at their structure, they have a lot of carbon in their chains. right? Those carbon atoms can be clipped 2 at a time, and they can feed into stage 2. And where do you make most of your energy? It's from stage 2 and stage 3. That's why fats give you a lot of energy, because there's a lot of 2 carbon fragments in fat. Here's the thing. You see this? It can be reversed. So if you have, and here's a neat little chart showing you this. If you have excess sugar, what happens to that excess sugar? So here's the glucose. Look, glucose can be converted into what? Fat. Or into glycogen. Proteins can be converted into amino acid, but they can also be converted into sugar. So we can convert these nutrients into other nutrients. So be careful when you, and we'll talk about this later on. I don't know if you know this, there's been a shift in the diet, or not a shift in the philosophy of the diet. When I was growing up, it was like, stay away from fat, right? Now it's changing. It's not so much stay away from fat, it's stay away from sugar. Sugar's a big evil. So if you pick up something that says it's fat-free, be careful. Guarantee it's probably full of sugar. And you can see from this that sugar can be converted into fat anyways, right? But we'll talk about this later on in the course. All right, ladies? That is it for respiration. The